let's take a look at protein structure. Of all the biological molecules, the most diversity that we have with regard to function lie with our proteins. So before we look at the specifics of what proteins are made out of, let's just take a look at all of the different functions that are present. Let's actually start by taking a look at something we've already mentioned, and that is looking at our cystic fibrosis protein. One of the jobs of proteins is to serve as transport inside of plasma membranes. So here we could see a protein that is structural. So it's this structure right here in blue. And here it's allowing substances like chloride ions to move across the membrane from inside of the cell to outside of the cell. We know that transport proteins sit in the cell membrane, the plasma membrane, and that sometimes these transport proteins are open, like the one here, and sometimes those transport proteins are closed and they won't allow something through. We also know that these proteins are really specific, meaning that it's not any protein that's going to allow these chloride ions through, it's just this particular one. If we're going to look at other substances moving across membranes, then we're going to see different proteins. So the one that moves sodium is a different one that moves chloride, and that's different than the one that transports glucose. Another function that proteins can have embedded in plasma membranes is acting as a regulatory protein. In this case, it's acting as a receptor. A receptor is something that's going to receive a signal from outside of the cell, which is out here, and it's going to set off a series of events inside the cell. So we notice that in the outside of the cell there are all sorts of signal molecules here, but it's only our orange triangles that are going to have the right fit and can actually bind with our receptor protein, number 2. Once that binding happens, then we are going to communicate inside of the cell a certain response. So maybe this right here is a hormone, which is a chemical that communicates with cells by attaching to receptors, and maybe the signal that we'll send to the cell is time for cell division. That might be a good example. Proteins can also function as enzymes. We've mentioned some things about enzymes this semester, and we'll talk a lot more about them as we go. But an enzyme is going to act as a catalyst. And a catalyst is something that's going to speed up or facilitate chemical reactions. So in this picture here, you can see that this purple thing is our enzyme, and it is a protein. And what's happening is that this substance right here, maltose, is binding with our protein, which is shown here in this panel. And the chemical reaction that occurred is that maltose was split into two things, glucose and another glucose. You'll notice that our enzyme here in purple didn't change at all, and that means that this enzyme can be used again. So that's another important function of proteins, is to act as enzyme. Another function of proteins is involved in muscle contraction. When we look at a muscle that is relaxed before contraction, we can see all of these proteins overlap slightly, and so each one of these are a different type of protein. However, when contraction takes place, then you can see these proteins overlap to a much greater degree. So you can think about your biceps muscle and your upper arm. When it's relaxed, when your elbow is straight, then you'll be able to see these fibers looking like this. However, when we look at the muscle when your elbow is bent, we would see that it looks a lot more like this. So some of these proteins are going to have these types of structure that allow them to be able to allow for contraction. Proteins can also be protective. One example is an antibody. Antibodies are not cells. Antibodies are proteins. And what you'll notice is this antibody is very specific. Out here on the end of this Y-shaped protein, there's a place where foreign invaders can bind, and that's called the antigen binding site. And so if you've had a cold recently, then you've produced antibodies that are specific to that particular area. So as you can see, there are many different types of proteins and many different functions. Different videos and different textbooks will classify them into different groupings. It's more important to me that you know the diversity that we have rather than the specifics of those categories. Now that we know something about the many different types of proteins, we need to zoom in and find out what these proteins are made out of, and how is it that we can have so many different types of proteins when we actually have just a few of the building blocks that make them up. If you remember back to our discussion of DNA, we said that DNA was composed of small subunits or building blocks that bonded together to make the larger structure of DNA. And we used the term monomer to refer to a single unit, and that we built those into large polymers. We're going to use similar terms here when we're talking about proteins. When we're looking at proteins, the building blocks are not nucleotides like we had in DNA, but instead our building blocks are amino acids. And our polymer then is going to be our final protein once we put those amino acids together. So I'd like to take a look at the structure of a single amino acid, and then we're going to put them together to make larger proteins. What we can see in this picture here is a generic structure of an amino acid. So I want to talk about the amino acids. There are 20 different amino acids. Don't worry, you don't have to know the names of all of these, but we'll learn the name of some as we go. All 20 amino acids have some things in common. So let's find out what they are. All 20 amino acids have a central carbon in the middle. They all have an amino group to one side, and this amino group contains nitrogen. Remember, nitrogen is one of those big four elements that organisms have a lot of, and there's a lot of nitrogen that's present in proteins. All 20 amino acids will have a hydrogen attached to that central carbon. And then all 20 amino acids will also have what's called a carboxyl group. If we notice, we have a carbon that's going to be double bonded to an oxygen plus an additional OH group. So sometimes you'll see this written like this, and sometimes we'll refer to this as a Cu group. So when you look at any amino acid, you should be able to identify everything that's in green here: the central carbon, the amino group, the carboxyl group, and that hydrogen. What makes all these 20 amino acids different from one another then is what's in this position called the R group. This is just a placeholder in this generic picture for what could be there. So let's actually take a look at a number of different amino acids and find out how they're different from one another in this R group. At the top of this figure, we'll see the exact picture we saw on the previous slide. We recognize that everything here is going to be identical between all of our amino acids. So of our 20, let's just take a look at 4. Alanine will contain everything that we've just talked about. So will valine. So will lysine. And finally, aspartic acid. But what's different about all of these amino acids? It's what is in this R group position. So alanine, for example, has in that position a CH3, some carbon and hydrogen. Valine also has carbon and hydrogen, and you can see there's actually 3 of them. Lysine in its R group has some more CH2, 4 of those, plus an additional group that has nitrogen in it. And you can see aspartic acid has some CH2 and another Cu group. Other type of amino acids might have sulfur inside of their R group. The point is that it's the R group that's going to make each of these amino acids different from one another, and it's the R group that's going to give each amino acid its particular character or property. Well, what does that mean? This is going to become important a little bit later, but we know that maybe some of these groups are going to be polar or not polar. That's going to determine whether this amino acid will mix with water or not. We also know that some of these R groups may have positive charges or negative charges, and that could influence how they interact with one another when we look at protein structure. All right, well, let's start to take some amino acids and put them together in the larger structure of a protein. When we start to join amino acids together, we start to form bonds in between the amino acids. I want to just show you a simplified picture. Let's say this is an amino acid, and we're simplifying what we saw on a previous picture. And we have another amino acid, and we want to start to link them together with a bond. The bond that holds together 2 amino acids is called a peptide bond. And then if we want to link another amino acid in a chain, we will put in another amino acid and hold it together with a peptide bond. These peptide bonds are covalent bonds. They're just a very specific type of covalent bonds. These are really strong bonds that are going to hold these together. And so as we're looking at how we take these individual monomers, amino acids, and link them together into larger proteins, our polymer, we'll find that from time to time we'll have 300, 400, 1,000 amino acids linked together to make these very large proteins. If we are to look inside of living cells, however, we're not just going to see amino acids linked together. We're actually going to see these really three-dimensional, dynamic, very complex proteins. So we need to look at the many levels of protein structure and how we go from linking amino acids together to ultimately making these very large and complex proteins. Protein structure involves 4 different levels. So the level that we've just put together is what we call our primary protein structure, and that's simply going to be the order of the amino acids. Here we can see in primary protein structure individual amino acids linked together, and I hope we remember then that all of these bonds holding every single circle together is a peptide bond. So if we want to know what the primary structure of a protein is, what we're really just asking is, what is the sequence of the chain of amino acids? Now remember, there's only 20 different amino acids, but these 20 different amino acids can be repeated over and over. We can put them in a different order, and we can end up then with thousands, tens of thousands, hundreds of thousands of different proteins from just 20 different kinds of amino acids. I use the analogy as our alphabet for this. We only have 26 letters in the alphabet, but think how many different words we can make because the length of the word varies as well as the order of the letters. And it's the same way here when we look at primary protein structure. From our primary structure, we can move on to secondary structure. And secondary structure is where we start to add some dimension. There are 2 types of secondary structure, and let's just focus on one, the one that we'll see the most, and that's called the alpha helix. It's essentially just taking a amino acid polypeptide, our primary structure, and starting to put it in a helix, similar to what we had in DNA. Now we know that this has to be stabilized, so oftentimes we're going to see hydrogen bonds. We remember those, hydrogen bonds. Hydrogen bonds are going to stabilize this helical structure. A good analogy for an alpha helix is when phones had cords, if you remember those, and you can think about how that phone cord would coil, and that's a great analogy for an alpha helix. Other proteins have what we call beta pleated sheets, where rather than a double helix, we'll instead see a folding pattern, and that is making a sheet rather than a helical shape. So we can see both types representing secondary structure. As we move from secondary structure to our third level structure, the term we use for third level is called tertiary protein structure. This tertiary protein structure is when our alpha helix now will start to fold, and it's going to fold in a very specific way. This is not random folding. There are rules according to folding, and it goes back to the order of our amino acids. We know that then when we fold this, we'll start to see this big knot structure. And then in there, we can also see that we've got an alpha helix here, or we might see a beta pleated sheet over here, showing those folds. Finally, we can make it to our fourth level protein structure, and that is quaternary structure. And this is when we'll actually take more than one of our third levels and put them together. So maybe the big blob that we have here, our big knotted structure on tertiary level, is just this lighter colored one here, and that we're going to get another one and put it over here for that darker color together. Now we have a very large protein that is at this quaternary protein structure level, and now we might call this hemoglobin or insulin. And when we actually look at those proteins, we can see that they're very large, and we're really looking at them at this quaternary level. The picture that we've just looked at is out of your textbook readings, but I wanted to just show you another picture out of your course manual that has a little bit more detail. Again, here we can see our primary structure with a little bit more detail about our amino acids. So here's an amino acid, and here's another one, and here's another one being linked together. So we can see that as they're linked together with peptide bonds, we're forming our polypeptide strand. We then can see our secondary structure of an alpha helix, and again we can see now more clearly those hydrogen bonds in dots here that are holding those together. And then how many of these could come together also to make another type of secondary structure, our beta-pleated sheet. Our tertiary structure here is represented by all of the folding that we talked about. And then finally, if we put more than one of these together, this one actually has 4 put together, we can see our entire fully functional protein. As we have learned more and more about proteins, we've been able to utilize a lot of technology and computer graphics to be able to help us understand the structure of proteins. And so I just wanted to show you a couple of them. You can see here the hemoglobin in a much more detailed way, created by a particular type of software that helps to show our alpha helixes and also a good deal of the folding that goes on. So here you can see the complexity of the hemoglobin. Hemoglobin actually has 4 polypeptides, 1, 2, 3, 4, put together to make the overall hemoglobin molecule. Insulin is another type of molecule, and there's 2 pictures here, one that shows this version and then one that shows this version of an insulin molecule. And again, when you look very closely, you're able to see some of the alpha helices that are here, and then in some cases, beta-pleated sheet. The final protein that I wanted to look at was the one that we're most interested in this case, and that is our cystic fibrosis protein. Remember that our cystic fibrosis protein is a transport protein that sits in the plasma membrane of our cells. And if we were actually to take that transport molecule and look at it up close, this is what we would see. Very complex structure with beta-pleated sheets, with alpha helices. And if we were to go back and look at this very specifically, we would be able to find the order of the amino acids. We know that along any strand of this, we could look at the order of the amino acids and be able to see primary structure as well. So keep in mind, while we're talking about taking amino acids and putting them together to build these large proteins, when we're talking about their functions, we're really looking at the large-scale quaternary structure so that we can understand how their shape actually contributes to their function. Proteins are one of the main reasons why your body needs to maintain homeostasis. When we look at proteins, we realize that they require a very specific environment in which to maintain their shape and therefore maintain their function. So we can see here an example of a protein at quaternary level. We also know that if we put this protein in an environment that it doesn't like adding heat or making the pH very acidic, our proteins are going to change their shape. What you can see here as we've moved from this picture to this picture is that our protein has started to unravel. We've started to lose some of our structure. And this is termed denaturation. Denaturation is when proteins lose their nature. They start to lose their shape. And when they do this, then their function is no longer possible. They will lose their ability to act as a transport protein, be a regulatory protein, act as a receptor. They won't be able to function as enzymes anymore. So this is in part why maintaining pH homeostasis, why maintaining a relatively stable body temperature for some organisms is so important. We'll talk more about the role that proteins have throughout our semester, but hopefully as we move forward in our cystic fibrosis case, we'll appreciate a lot more about what this protein looks like.